Hi, I'm Alexis Reymbaut and I'm Head of Research and Product Development at Random Walk Imaging. I'm going to talk to you about multidimensional diffusion MRI, or MDD. Let's start with a quick refresh on how diffusion relates to tissue microstructure. If I consider an empty diffusion medium and I drop some ink within it, after a few hundred milliseconds there's going to be a diffusion pattern that's going to form. And this diffusion pattern has a certain size and a certain shape. If I add obstacles to the diffusion process, then this diffusion pattern is going to get resized and reshaped accordingly. So for example here, with these isotropic cells that are densely packed, this diffusion pattern is going to reduce in size proportionally to the inverse cell density. And here, if you have these elongated cells as obstacles, then you see that the diffusion pattern gets reshaped to fit the anisotropy of the underlying cells. In the brain, these relate to white matter, gray matter in a simplified manner, and CSF, cerebrospinal fluid. In this cellular profile of a glioma, you see that the cells are roughly isotropic and they only vary in terms of cell density. So if I try to match diffusion patterns on top of this image, I would get something like this, where you have small diffusion patterns in areas of very high cell density and big diffusion patterns in areas of low cell density. In this cellular profile of a meningioma, you see that instead the cells tend to be more elongated, and then the diffusion profiles would have certain anisotropies that would match those. Now let's address the main issue that diffusion MRI may face, sub-voxel heterogeneity. In most diffusion MRI voxels, you will find a collection of distinct diffusion patterns or diffusion tensors if we talk about the mathematical objects that represent them. So if I take this coronal slice of the brain imaged by diffusion MRI and I isolate one voxel, in this voxel you may have a collection of environments. So for example, in this case, the voxel was close to the ventricle, so you may expect some CSF. And it was also in a voxel that contains some white matter, so you may find these white matter bundles. And you see that the size of the voxel is pretty much millimetric, while the size of cells is pretty much micrometric. So you can expect to have a large variety of cells. And then, if you want to describe these using diffusion patterns or diffusion tensors, you will need a collection of them for any given voxel. The problem then becomes, how can you identify the features of such collections? One way to do it is to use trace diffusion weighted imaging, or trace WI. In this case, for example, you can acquire an image with a magnetic gradient aligned with this X direction, which is usually the left-right direction, like here. And you see that in this image, for example, the corpus callosum has disappeared because it is mostly aligned with this XY direction, at least for the medial part of the corpus callosum. And then given these fibers are aligned with the gradient, they will yield a very minimal signal. And then you can acquire another image with a gradient aligned with the Y direction, which is this anterior-posterior direction, and then you see that the optical tracks are dark. And finally, you can acquire an image in the Z direction, which is the inferior-superior direction, and then you see that the corticospinal tracks tends to be dark. Besides this one, you can acquire one image that doesn't have any diffusion gradient on, which is called the S0 image, and then you can define this DWI image as the cubic root of Sx, Sy, Sz, and it's related to this S0 you acquired with this exponential factor. So you see that compared to S0, the diffusion weighted image has a lower signal, and this lower signal is given by the B value, the strength of the diffusion encoding, and this ADC, apparent diffusion coefficient. It is inversely related to the voxel averaged cell density. Diffusion tensor imaging, or DTI, goes a bit beyond. It captures a single average diffusion tensor per voxel, and then the signal in DTI writes as the non-diffusion weighted signal S0, weighted down by this exponential that depends on the B tensor, or B matrix, and the average diffusion tensor for the voxel that is considered. This is the Frobenius inner product between the two tensors. Let's illustrate this concept in this voxel that contains two fiber populations, the blue and the orange ones. If I consider the average diffusion tensor, In this voxel, it would look like this. It's a tensor that is mostly aligned vertically because, in average, the two fiber populations are aligned vertically. Diffusion-wise, you see that the average diffusion tensor has some anisotropy, but this anisotropy is lower than that of each of these fiber populations. The reason behind this is because you're averaging the voxel content. So in average, there is a lot of diffusion happening vertically, but there's some diffusion happening horizontally, at least in this horizontal plane, because of the crossing. So then in average, it looks like a fatter tensor. From this average diffusion tensor, DTI computes two metrics called the mean diffusivity, linked to the trace of the tensor, and the fractional anisotropy, linked to the anisotropy of the diffusion tensor. You see that the mean diffusivity lights up in the ventricles or in the water around the brain because they are more diffusive than the rest. And the fractional anisotropy lights up mainly where you have these fiber populations, white matter fiber populations. However, you see that you have these areas of massive crossings between fiber populations where the fractional anisotropy goes down. The reason being that in these areas of crossing, the voxel content looks isotropic in average because, again, the voxel is quite big compared to the cellular structures within. In other words, the fractional anisotropy does not capture white matter integrity. Diffusion kurtosis imaging, or DKI, has been developed later than DTI in the hope of capturing for the first time some notion of complexity of the tissue or heterogeneity of the tissue. DKI looks a lot like trace DWI but extends it. And it captures this first measure of tissue heterogeneity called the mean kurtosis. So here the signal for DKI looks a lot like trace DWI, this S0 exponential minus b times the apparent diffusion coefficient. But you see this second term in the exponential in b squared linked to the ADC squared and this mean kurtosis k squared divided by 6. You can also write the logarithm of the signal DKI divided by S0, and you see that you have these two cumulants that show up. This is a two-term cumulant expansion. The first cumulant is minus the ADC, and the second cumulant is ADC squared k squared over 6. The problem with the mean kurtosis is that it's not specific. For instance, you could have two different sources of heterogeneity that would contribute the very same way to the mean kurtosis k. In this case, for example, you see this voxel content that contains varying cell densities that are shown with these diffusion patterns with varying sizes. And here you have this other voxel content that contains elongated diffusion patterns. Conventional diffusion encoding techniques suffer from an inherent lack of specificity. And this lack of specificity is due to two things. The first one is that all the previous techniques I mentioned rely on certain assumptions. And these assumptions may and will, in this case, have limiting consequences. The other reason is that conventional diffusion encoding techniques rely on conventional gradient waveforms like PGSE gradients, post-gradient spin echo, that will yield poor information. And that's where multidimensional diffusion MRI comes into play. The point of multidimensional diffusion MRI is not to replace the previous conventional diffusion encoding, but just to complement it with other types of encoding. This enables to acquire complementary pieces of diffusion information that yield a more refined picture of the voxel content. For example, you can still consider this post-gradient spin echo sequence, which we name now linear encoding, for the reason that this sequence will sensitize the signal to the diffusion happening along its acquisition direction, hence linear encoding. There's another kind of encoding that can be defined in the framework of multidimensional diffusion MRI. It's planar encoding. In this case, you see the different colors that relate to orthogonal axes in space. Planar encoding sensitizes the diffusion signal to diffusion happening perpendicular to the acquisition axis. So in this plane that is perpendicular to the dashed axis. Finally, you could define, for example, spherical encoding, which is a bit more complicated in terms of waveform, but yields an encoding that is sensitizing the diffusion signal to any isotropic diffusion patterns. Why does it work? Let's investigate this using this example. In this example, we face again the two voxel contents I previously mentioned. This one contains diffusion patterns or diffusion tensors with varying sizes. In this voxel content, you have anisotropic diffusion patterns or diffusion tensors that are randomly oriented, which could be the case of a meningioma, for example. If you use linear encoding to study the first voxel content, you will be sensitive to these high diffusivities in the big diffusion tensors and to small diffusivities in the small diffusion tensors. And then the diffusivity distribution that you will probe will have a peak at slow diffusivities and one peak at fast diffusivities. If you use the same encoding to study this voxel content, and for this purpose I will copy and paste the axis of acquisition of this specific linear B matrix or B tensor, you will be sensitive to the high diffusivity along the acquisition axis for these blue tensors, but also to the small diffusivities acquired across the red tensors. So the diffusivity distribution that you will probe will look a bit like this, where you have still this contribution at low diffusivity and this contribution at high diffusivity. And in practice, these two distributions share the same average and the same variance, which will make very difficult to tease them apart based on these properties of the diffusivity distribution. This is why the mean kurtosis of DKI cannot tease them apart. Instead, if you use spherical encoding, the first voxel content will yield the very same diffusivity distribution in the signal. There will be no difference whatsoever because all the voxel content diffusion-wise is isotropic. However, if you use spherical encoding on the second voxel content, it will sensitize the diffusion signal to isotropic diffusion patterns. It happens that all of these anisotropic diffusion tensors share the same isotropic component or the same trace. In this case, you can see that the diffusivity distribution will be extremely peaked around that value of diffusivity that is being probed. And then if you combine these two pieces of information, you can say for sure that if you observe the same distribution with linear and spherical encoding, then you're facing a voxel content that is purely isotropic. And if you compare the two encodings in the second voxel content and you see that one of these yields this kind of distribution and the other one is extremely peaked, then you can infer that the voxel content is made of anisotropic diffusion tensors that may or may not be aligned, but at least share the same trace. A good in vivo application of this principle is to tease apart glioma and meningioma only using diffusion MRI. The reason is that gliomas tend to have isotropic cells that are packed with different cell densities, and meningiomas tend to have these anisotropic cells that most of the time are quite randomly oriented on the voxel scale. This is some work from Filip Stepankevich et al. That studies meningioma and glioma from the standpoint of diffusion MRI alone. In these maps you see the mean kurtosis from DKI, and you see that the contrast within the meningioma and the glioma is identical in these two tumors. But using linear and spherical encoding and other approaches that are enabled via multidimensional diffusion MRI, you can obtain new metrics. For example, here you have this anisotropic mean kurtosis, MKA, and this isotropic mean kurtosis, MKI. And the mean kurtosis anisotropic is sensitive to microscopic anisotropy and is specific to it. And you see that it only lights up in the meningioma, but not in the glioma. The reason being that anisotropic cells are only existent in the meningioma. It's important to understand that microscopic anisotropy does not care whether or not anisotropic cells are aligned or not over the voxel scale. Microscopic anisotropy is very different from fractional anisotropy, which is confounded by this orientational effect. The isotropic mean kurtosis, MKI, is sensitive to diffusion variance, which is the variance of isotropic diffusivities, or the variance of cell densities from a biological standpoint. You see that this metric lights up in the glioma but not in the meningioma, because meningiomas tend to have roughly uniform cell densities, while glioma do not. Another important contribution from multidimensional diffusion MRI is that anisotropy can be seen from this macroscopic and microscopic points of view. For example, FA has a macroscopic point of view because it computes anisotropy once averaged over the entire voxel scale, hence the problems that are here in these green circles. On the other hand, microscopic fractional anisotropy is a metric that can quantify microscopic anisotropy. And you see that microscopic FA does not vanish in areas of crossing, because it is not sensitive to the alignment of cells over the voxel scale. And you see also that this micro-FA is not zero in gray matter, because it is known that gray matter features some degree of microscopic anisotropy. This orientation and order parameter OP measures cell alignment over the voxel scale. And you see that these dark spots that were to be found before in the fractional anisotropy are now here in this orientation order, which shows that the fractional anisotropy is actually confounding these two contributions. As a proof of concept of the power of microscopic anisotropy is an application in multiple sclerosis that was recently performed in Denmark. The point of this study was to compare fractional anisotropy and microscopic fractional anisotropy in MS lesions to see whether or not microscopic anisotropy, unlike FA, would correlate with clinical scores in MS. Let's look at the main result. This graph shows fractional anisotropy as a function of different parameters to assess whether or not correlations exist between them. The two columns correspond to the two brain hemispheres. The rows correspond to the parameters that may or may not be correlated with fractional anisotropy. You have H, EDSS, Expanded Disability Status Scale, which quantifies clinical disability, and SMDT, Symbol Digit Modalities Test, that quantifies cognitive processing. Finally, the dots are blue for healthy controls, filled orange for relapsing remitting MS patients, and empty orange for primary progressive MS patients. You can see that the p-values associated with correlations between fractional anisotropy and either EDSS for clinical disability or SMDT for cognitive processing are quite high, which shows that FA does not correlate with these scores. On the other hand, microscopic fractional anisotropy does present much lower p-values. This reflects the fact that microscopic fractional anisotropy actually quantifies white matter integrity. Thank you very much for your attention.